Now let's start with physiology portions, human physiology. In this video, we are going to start the initial portion that is related to the gastrointestinal system and nutrition, then the excretory system. Then after some time, we will be learning about the respiratory system, nerve system, major systems related to our human body. Then the special senses such as vision, structure of eyeball, fluid system of the eye, topics related to eye, and the formation of image on the retina. And then the tactile response, then the cardiovascular system in detail along with lymphatic system, then environmental physiology like thermal regulation, comfort zone, normal body temperatures, and temperature regulating mechanism, and the role of hypothalamus, thyroid, and adrenal gland in this process, and the acclimatization process also. So in this physiology lecture, we are focusing on the organization of the gastrointestinal system. So what is the writing objective? List the different types of smooth muscles, outline the process of smooth muscle contraction, compare smooth skeletal and cardiac muscles and their functions, then try to illustrate the neural control of the gastrointestinal tract and outline the gastrointestinal blood flow. So before moving on to the complexity of this topic, let's learn the glossary of terms. What is enteric nerve system? A nerve system that the gastrointestinal tract has for its own functions. It lies entirely in the wall of the gut, beginning in the esophagus and extending all the way to the anus. So that is enteric nerve system. Then slow waves, slow undulating changes in the resting membrane potential and are not action potentials. They determine the rhythmical gastrointestinal contractions. Then spike potentials, true action potentials. They occur automatically when the resting membrane potential of the gastrointestinal smooth muscles become more positive than about negative 40 mV. Then syncytium refers to the multinucleated mass of cytoplasm that is not separated into individual cells. The alimentary tract. Now we are moving on to the major topic. Let's have an introduction. The alimentary tract provides the body with a continual supply of water, electrolytes, vitamins, nutrients, which requires movement of food through the alimentary canal or tract, secretion of digestive juices and digestion of the food, absorption of water, various electrolytes, vitamins, and digestive products, circulation of blood through the gastrointestinal organs to carry away the absorbed substances, then control of all these functions by local nerves and hormonal systems. In this chapter, we will discuss the organization and basic principles of functions in the entire alimentary tract, and in its subsequent chapters, the specific functions of different segments of the tract will be addressed. First, general principles of gastrointestinal motility. Physiological anatomy of the gastrointestinal wall. Which are the layers? The serosa, a longitudinal smooth muscle layer, a circular smooth muscle layer, a submucosa, and mucosa. In addition, sparse bundles of smooth muscle fibers, the mucosal muscle, lie in the deeper layers of the mucosa. The motor functions of the gut are performed by the diffusion layers of smooth muscles. So let's learn about the types of smooth muscle. For the sake of simplicity, smooth muscle can generally be divided into two major types, which are shown like multi-unit smooth muscle, then unitary or single unit smooth muscle. Multi-unit smooth muscle will be there, then single unit smooth muscle will be there. So multi-unit smooth muscle is composed of discrete separate smooth muscle fibers. Each fiber operates independently of the others and often is interviewed by innervated by a single nerve ending as occurs for skeletal muscle fibers. Next, unitary smooth muscle. Earlier we have learned multi-unit smooth muscle. Multi-unit smooth muscle is composed of discrete, separate, smooth muscle fibers. Each fiber operates independently of the others and often is innervated by a single nerve ending as occurs for skeletal muscle fibers. Next, unitary smooth muscle. Unitary smooth muscle is also called syncytial smooth muscle or visceral smooth muscle. It consists of a mass of hundreds to thousands of smooth muscle fibers that contract together as a single unit. The specific characteristics of smooth muscles in the gut are the following. Gastrointestinal smooth muscle functions as a syncytium. The individual smooth muscle fibers in the gastrointestinal tract are 200 to 500 mm in length and 2 to 10 mm in diameter, and they are arranged in bundles of as many as 1000 parallel fibers. In the longitudinal muscle layer, the bundles extend longitudinally down the intestinal tract. In the circular muscle layer, they extend around the gut. Within each bundle, the muscle fibers are electrically connected with one another through the large number of gap junctions that allow the low resistance movement of ions from one muscle cell to the next. Each muscle layer represents a branching lattice work of smooth muscle bundles and functions as a syncytium. That is, when an action potential is elicited anywhere within the muscle mass, it generally travels in all directions in the muscle. The distance that it travels depends on the excitability of the muscle. Also, because of few connections exist between the longitudinal and circular muscle layers, excitation of one of these layers often excites the other as well. So, electrical activity of gastrointestinal smooth muscle. The smooth muscle of the gastrointestinal tract is excited by almost continual flow intrinsic electric activity along the membrane of the muscle fibers. This activity has two basic types of electrical waves, slow waves and spikes. In addition, the voltage of resting membrane potential of the gastrointestinal smooth muscle can change to different levels, which can also have important effects in controlling motor activity of the gastrointestinal tract. We just try to learn the structure of alimentary tract. The alimentary tract begins at the mouth and is made up of discrete areas that aid the digestion and absorption of nutrients. Each area in the tract contributes to the efficient processing of the nutrients. This include the GI associated organs such as liver, pancreas, which provide important secretions including bile, enzymes and buffers, and some other parts. So first, mouth. Saliva. Lingual lipase will be there. Salivary, alpha amylase and R proteins are there. Then esophagus, electrolytes and mucus. Then liver and gallbladder, bile and buffers or electrolytes will be there. Then stomach, HCl, intrinsic factor, pepsinogens, gastric lipases, mucus, gastrin, somatostatin and histamine. Then pancreas contain exocrine glands, buffers and enzymes like trypsin, chymotrypsin, procarboxypeptidases, lipases, colipases, proelastase, alpha amylase, monitor peptide like trypsin inhibitor stimulates CCK, endocrine glands, insulin, glucagons, somatostatin. Then small intestine contain buffers, mucus and electrolytes along with enterokinase, brush border disaccharidases, brush border peptidases, secretin, gastrin, CCK, VIP, GIP, motilin, 5-HT, somatostatin and all those related things. So earlier we have learned about two types of waves like slow waves and spike potentials. So what are slow waves? Most gastrointestinal contractions occur rhythmically, and this rhythm is determined mainly by the frequency of so-called slow waves of smooth muscle membrane potential. These waves are not action potentials. Instead, they are slow undulating changes in the RMP. The intensity usually varies between 5 and 15 MV, and their frequency ranges in between parts of the human gastrointestinal tract from 3 to 12 minutes. About 3 in the blood of the stomach, as much as 12 in the duodenum, and about 8 to 9 in the terminal ileum. The precise cause of the slow waves is not completely understood, although they appear to be caused by complex interactions among the smooth muscle cells and specialized cells called the interstitial cells of Kajal, which are believed to act as electrical pacemakers for smooth muscle cells. These interstitial cells form a network with each other and are interposed between the smooth muscle layers with synaptic-like contacts to smooth muscle cells. The interstitial cells of Kegel undergo cyclic changes in membrane potential due to unique ion channels that periodically open and produce inward pacemaker currents that may generate slow wave activity. The slow waves by themselves usually do not cause muscle contraction in most parts of the gastrointestinal tract, except perhaps in the stomach. Instead, they mainly excite the appearance of intermittent spike potentials, which in turn actually excite the muscle contraction. So, what is spike potential? The spike potentials are true action potentials. They occur automatically with the RMP of the gastrointestinal smooth muscles become more positive than about 40 MV. Normal RMP in smooth muscle fibers of the gut is between 50 and minus 60 MV. The higher the slow wave potential raises, the greater the frequency of the spike potential becomes, usually ranging between 1 and 10 spikes per second. The spike potentials last 10 to 14 times as long in gastrointestinal muscles as the action potentials in large nerve fibers, with each gastrointestinal spike lasting as long as 10 to 20 milliseconds. Another important difference between the action potentials of the gastrointestinal smooth muscle and those of nerve fibers is the manner in which they are generated. In nerve fibers, the action potentials are caused almost entirely by rapid entry of sodium ions to sodium channels to the interior of the fibers. However, in gastrointestinal smooth muscle fibers, the channels responsible for the action potentials allow especially large number of calcium ions to enter along the smaller numbers of sodium ions, and therefore called calcium sodium channels. They are called calcium sodium channels. Okay, these channels are much more slower to open and close than are the rapid sodium channels of large nerve fibers, therefore increasing the duration of the action potentials. Also, the movement of large amount of calcium ions to the interior of the muscle fiber during the action potential plays a special role in causing the intestinal muscle fibers to contract, as we discussed shortly. So when you just look at the cross section of a gut, we can see serosa layer is there, longitudinal muscle, circular muscle along with submucosa, then mucosal muscles, mucosa, epithelial lining, mucosal gland, myendric nerve plexus, Meissner nerve plexus, and submucosal gland along with a mesentery. So these are the cross-sectional regions of a gut. Serosa is the outermost layer. Inside of that, we will be moving into the gut. So we will be first referring to the last outermost layer, that is the serosa, then longitudinal muscle, circular muscle, submucosa, mucosal muscle, mucosa, epithelial lining, then mucosal gland, myendric nerve plexus, Meschna nerve plexus, submucosal gland, and mesentery. Then just we try to learn the multi-unit and unitary smooth muscle. You can say multi-unit are just branch type, and then when we just look at the unitary smooth muscle, you can say it has adventitia, medial muscle fibers and endothelium, and small artery will be there. So next, changes in voltage of the resting membrane potential. In addition to the slow waves and spike potentials, the baseline voltage level of the smooth muscle RMP can also change. Under normal conditions, the RMP averages about 56 MV, but multiple factors can change this level. When the potential becomes less negative, which is called depolarization of the membrane, the muscle fibers become more excitable. When the potential become more negative, this is called hyperpolarization, and fibers become less excitable. Next, factors that depolarize the membrane, making it more excitable. Stretching of the membrane. Stimulation by acetylcholine released from the endings of parasympathetic nerves, and stimulation by several specific gastrointestinal hormones. Important factors that make the membrane potential more negative, that is, that hyperpolarize the membrane and make it less excitable, are the effect of norepinephrine or epinephrine on the fiber membrane, and stimulation of the sympathetic nerves that secrete mainly norepinephrine at their endings. Next, entry of calcium ions causes smooth muscle contraction. Smooth muscle contraction occurs in response to entry of calcium ions into the muscle fiber. These calcium ions, acting through a calmodulin control mechanism, activate the myosin filaments of the fiber, causing attractive forces to develop between the myosin filaments and the actin filaments, thereby causing the muscle to contract. The slow waves do not cause calcium ions to enter the smooth muscle fiber, they cause only entry of sodium ions. Therefore, the slow waves by themselves do not usually cause muscle contraction. However, it is only during the spike potentials that significant quantities of calcium ions do enter the fibers and cause most of the contraction. Next, neural control of gastrointestinal function. Enteric nerve system. This term we have already mentioned earlier. Enteric nerve system. The gastrointestinal tract has a nerve system at its own called the enteric nerve system. It lies entirely in the wall of the gut, beginning in the esophagus and extending all the way to the anus. The number of neurons in this enteric system is about 100 million, nearly equal to the number of the entire spinal cord. This highly developed enteric nerve system is especially important in controlling gastrointestinal movements and secretion. The enteric nerve system is composed mainly of two plexus, like an outer plexus lying between the longitudinal and circular muscle layers called the myenteric plexus or Overbach plexus. We will be learning about it later. Myenteric plexus. Then an inner plexus called the submucosal plexus or Meschna plexus, which lies in the submucosa. The nerve connections within and between these two plexus are very important. The myenteric plexus controls mainly the gastrointestinal movements, and the submucosal plexus controls mainly gastrointestinal secretion and local blood flow. Extrinsic sympathetic and parasympathetic fibers that connect to both the myenteric and submucosal plexus. Although the enteric nerve system can function independently of the extrinsic nerves, stimulation by the parasympathetic and sympathetic nerves can greatly enhance or inhibit gastrointestinal function, as we discussed earlier. Also, the sensory nerve endings that originate in the gastrointestinal epithelium or gut wall and send afferent fibers to both plexus of the entire system as well as to the preverbal ganglia of the sympathetic nerve system. to the spinal cord and in the vagus nerve all the way to the brainstem. The sensory nerves can elicit local reflexes within the gut wall itself and still other reflexes that are relayed to the gut from either the preverbal ganglia or the basal regions of the brain. Differences between the meanderic and submucosal plexus have been given. So, comparison of smooth skeletal and cardiac muscle, when we just look at that, like in the case of shape of muscle fiber, smooth muscle is fusiform, but skeletal muscle is cylindrical and cardiac muscle is branched one and attached to bone. Smooth muscle is not attached to bone, but skeletal muscle is attached, but cardiac muscle is not attached. Mitochondria is less in case of smooth muscle, but few in case of skeletal muscle, many in case of cardiac muscle. Sensation is seen in single unit smooth muscle, but contracts as a sensation in case of cardiac muscle, but skeletal muscle it's not present. Then sarcotubular system is poorly developed in case of smooth muscle, but it's well developed in skeletal muscle and it's moderately developed in case of cardiac muscle. Then contraction speed is very slow in case of smooth muscle but very fast in case of skeletal muscle, but it's slow in case of cardiac muscle. Then electrical spread between fibers is possible through gap junctions in case of smooth muscle. It's absent in the skeletal muscle but possible through intercalated discs and gap junctions in case of cardiac muscle. Then resting membrane potential is minus 50 in case of smooth muscle, minus 80 in case of skeletal muscle and minus 90 in case of cardiac muscle. Then refractory period is long in case of smooth muscle and cardiac muscle, but it's very short in case of skeletal muscle. So the sensory nerve endings that originate in the gastrointestinal epithelium or gut wall and send afferent fibers to both plexus of the enteric system as well as to the prevertebral ganglia of the sympathetic nerve system, to the spinal cord and in the vagus nerves all the way to the brainstem. The sensory nerves can elicit local reflexes within the gut wall itself and still other reflexes that are relayed to the gut from either the preverbal vertebral ganglia or the basal regions of the brain. Differences between the meanderic and submucosal plexus have been given, and the types of neurotransmitters secreted by enteric neurons. So when we just look at the structure of neural control of the gut wall, the meanderic and submucosal plexus are there. So the extrinsic control of these plexus by the parasympathetic and sympathetic nerve system is there, and sensory fibers passing from the luminal epithelium and gut wall to the enteric plexus and then to the preverbal ganglia of the spinal cord and directly to the spinal cord and brainstem is also very important. So sympathetic and parasympathetic, mainly postganglionic and but post-sympathetic in case of preganglionic. Then meanderic plexus will be there, submucosal plexus, epithelial region, sensory neurons to prevertebral ganglia, spinal cord and brainstem. So we were learning about the types of neurotransmitters secreted by enteric neurons. In an attempt to understand better the multiple functions of the gastrointestinal enteric nerve system, researchers have identified a dozen or more of different neurotransmitter substances that are released by the nerve endings of different types of enteric neurons. Autonomic control of the gastrointestinal tract. Parasympathetic stimulation, so which are they? Acetylcholine will be there, norepinephrine, adenosine triphosphate, serotonin, dopamine, substance P, cholecystokinin, vasoactive intestinal peptidase, somatostatin, leukenkephalin, leukenkephalin, and metenkephalin. Then bombacin. The specific function of many of these are not well known enough to justify discussion. I have to point out the following. Acetylcholine most often excites gastrointestinal activity. Norepinephrine and epinephrine almost always inhibit gastrointestinal activity. So when I just learn about the parasympathetic stimulation, increase activity of the enteric nerve system. The parasympathetic supply to the gut is divided into renial and sacral divisions, and except for a few parasympathetic fibers to the mouth and pharyngeal regions of the alimentary tract, the renial parasympathetic nerve fibers are almost entirely in the vagus nerves. These fibers provide extensive intervention to the esophagus, stomach, pancreas, and somewhat less to the intestines down through the first half of the large intestine. The sacral parasympathetics originate in the second, third, and fourth sacral segments of the spinal cord and pass through the pelvic nerves to the distal half of the large intestine and all the way to the anus. The sigmoidal, rectal, and anal regions are considerably better supplied with parasympathetic fibers that are the other intestinal areas. These fibers function especially to execute the defecation reflexes. The postganglionic neurons of the gastrointestinal parasympathetic system are located mainly in the myenteric and submucosal plexus. Stimulation of the parasympathetic nerves cause general increase in activity of the entire enteric nerve system, which in turn enhances activity of most gastrointestinal functions. Next, sympathetic stimulation usually inhibits gastrointestinal tract activity. The sympathetic fibers to the gastrointestinal tract originate in the spinal cord between segments T5 and L2. Most of the preganglionic fibers that innervates the gut after leaving the cord enter the sympathetic chains and lie lateral to the spinal column, and many of these fibers then pass on through the chains to outlying ganglia such as to the celiac ganglion and various mesenteric ganglia. Most of the postganglionic sympathetic neuron bodies are in these ganglia, and postganglionic fibers then spread through postganglionic sympathetic nerves to all parts of the gut. The sympathetics innervate especially all of the gastrointestinal tract rather than being more extensive nearest the oral cavity and anus, and is true for the parasympathetics. The sympathetic nerve endings not only secrete mainly norepinephrine. In general, stimulation of the sympathetic neuron system inhibit activity of the gastrointestinal tract, causing many effects opposite to those of the parasympathetic system. It exerts its effect in two ways. To a slight extent, by direct effect of secreted norepinephrine to inhibit intestine tract smooth muscle except the mucosal muscle, which it excites, and to a major extent by an inhibitory effect of norepinephrine on the neurons of the entire enteric nerve system. Strong stimulation of the sympathetic system can inhibit motor movement of the gut so greatly that this can literally block movement of food through the gastrointestinal tract. Next, afferent sensory nerve fibers from the gut. Many afferent sensory nerve fibers innervate the gut. Some of the nerve fibers may have their cell bodies in the enteric nerve system and have some of them in the dorsal root ganglia of the spinal cord. The sensory nerves can be stimulated by irritation of the gut mucosa, excessive distension of the gut, or the presence of specific chemical substances in the gut. Signals transmitted through the fibers are then cause excitation, or under other conditions inhibition of intestinal movements or intestinal secretion. In addition, other sensory signals from the gut go all the way to multiple areas of spinal cord and even the brainstem. For example, 80% of the nerve fibers in the vagus nerve are afferent rather than efferent. The afferent fibers transmit sensory signals from the gastrointestinal tract into the brain medulla, which in turn initiates vagus reflex signals that return to the gastrointestinal tract to control many of its functions. Gastrointestinal blood flow. Splanchnic circulation. The blood vessels of the gastrointestinal system are part of a more extensive system called the splanchnic circulation. It includes the blood flow through the gut itself plus blood flow through the spleen, pancreas and liver.